Hey all, hope you're doing well. Here is the video I promised you about breaking down the mock exam FRQ question number 1. So we're going to go ahead and start with this digital annotation guide that you can see in front of you. I'm going to share this link with all of you. It is on Google Drive. So the first thing that you're going to do, I'm going to kind of run through this and I'm going to show you how I want you to do this in your notes. And again, you guys are all taking notes on the strategy, okay? So maybe you're mocking this up in your notes like this. The way that I'm going to do it in a second. So you're not writing down, you don't necessarily have to write down all of these things. You guys will get this document. But I do want to just review it really quickly before I show you how to use it. So the first thing that you're going to do is obviously read the prompt. Okay. The second step is that as you're reading, you're going to record 3 maximum words to look up and then actually look them up once you're done reading the prompt, okay? You're going to do the same thing with processes and biological concepts. The reason for this is you guys are going to... you guys are only going to have a limited amount of time, so you don't want to be looking for every single thing that's in the prompt. You want to pick, okay, what do I think are the most important words to actually look up and processes to look up. And let me quickly jot down those notes so that it can help me to actually fill out this important information. So the fourth step, so this shouldn't take a whole lot of time. If you feel like you're good and you don't have to look anything up, you can literally just like not do these, okay? That's fine. And then so you could just not do these if you don't want to. But if they help you to understand the prompt, you should be looking those up, okay? I don't want us to spend more than about 2 minutes on this, so I'm going to show you guys how to do that in a second. Your step 4 is you're going to then write down important information about the prompt, okay? So this can be key points about the experimental design or biological information. So this could be like IVDV control, right? This could be like if you have a transcription question, maybe you're jotting down information that you actually already know. Okay, so that's what your relevant outside information you already know or that you looked up. That could be important. Also, just a summary of what's happening. Like this shouldn't be more than a couple minutes. I don't want you to take like expansive notes, but I do want you to have some information down that you're picking out that's important. Okay. And then step 5 is going to be reading the actual questions and filling out this table. I'm going to show you guys how I want you to do this in your notes. You're not going to make all of these like things in your notes. Like you're not going to put all of the words that are here, but you are going to have your question letter kind of as a T-chart. So I would put A, and then maybe I'd write predict, right? Which I know is just 1 is the level of detail, and then justify, right, is a 3. So I know I need to, maybe I want to remind myself to bring in evidence, right? So that's what will go here, and then you'll write down any detail here. And then I would write B, same thing, and I'd write my detail here, okay? The good thing about this section that I'm showing you guys here is that if you write down an initial response, even if it's notes and you don't get a chance to actually type it out, you can take pictures of this once you get to your upload time. You can take pictures of this and actually upload this, okay, before you actually do your final. So if you're running low on time, that's a really good time saver as well. Okay, cool. So I'm going to go ahead and this, okay, and what we're looking at with this, which is that we have these steps to kind of break down these questions. And again, the rationale for this is that I want you guys to have a how-to on these digital questions. And I know we're practicing like this. This might not be the best thing for you. Maybe you're feeling like it's slowing you down or it's harder. I do want you to try it today, and I'm going to have you upload your notes in the final task. So make sure that you take a look at this, the separate link that I'm going to post as a resource as well. And best of luck. Please see your feedback. I said this in the other video. Please see your feedback on this FRQ before the assessment on Friday. I'm going to give you feedback both on your notes and on the actual answer to the question. That's all I got. Have a great day, guys. Move on to the actual question. So if you guys remember, this was the question from the mock exam. Okay, this was question number 1. I am going to go through this. I'm going to read it, and I'm actually going to fill out a very similar template to what I just showed you. Okay, so I'm going to go ahead and start. And remember, you're not going to be able to actually annotate what's on the test, which is why we're doing this. So I just want to remind you guys that like I'm not going to actually mark up this question that we're going to do parts A, B, and C for. I'm just going to put this into my notes. So the first thing that I'm going to do is just notice that there's a figure here, okay, model of a 2-step enzymatic plant pathway for synthesis, okay? So, excuse me, auxins, I'm already annotating because, you know, I'm just so used to that. Auxins are plant hormones. Let me do this so that I can show you guys where I'm at. Are plant hormones that coordinate several aspects of root growth and development. IAA, so indole 3 acetic acid, is an auxin that is usually synthesized from the amino acid tryptophan. So as I'm reading through this first sentence even, right, there's a lot of words that I might not know, right? I might not understand what auxins are, okay? It is clear that you don't need to know what auxins are because they tell you they're plant hormones, okay, that coordinate several aspects. Maybe you really don't understand what a hormone is. Maybe you remember a little bit, right? But if you feel like you need to look up hormone because that is like impeding you, it's stopping you from understanding what this sentence says, look up hormones, right? And I also am noticing, right, that I also have this indole acetic acid is an auxin. It's usually synthesized from the amino acids tryptophan. There may be other words that I don't know here, but I do want to emphasize that like you should only be looking up words that are words that we've already talked about this year that maybe you don't remember, okay? You should not be looking up indole acetic acid and spending time looking up this word, right? They tell us IAA is an auxin that's usually synthesized. They're giving us information about it, and it's also in this. So we're trying to figure out what it is. The purpose of this is not to look up what this thing is. The purpose is for us to figure out what this system is, which we'll see in a second. So we're not looking up specific amino acids, things like that. Maybe I don't remember what amino acid is, and maybe I don't remember what synthesized is. Okay, remember we want to try to keep it, we want to try to keep it down to about 3. Doing this. Cool. And then the next sentence tells us that the gene, okay, encodes an enzyme that converts tryptophan to this I3PA, indole 3 pyruvic acid, which is then converted to IAA by an enzyme encoded by the gene YUC, okay? So again, there are a lot of words that we've never seen before, that some of these like I hadn't seen before I looked at this question, right? There's specific molecules. The purpose of this is not to know what those specific molecules are. It's to use the information we're given to kind of hypothesize that. And you probably aren't going to be able to Google whatever they give you. You will not be able to Google it, right? They're not going to give you something that's Googleable. So keep that in mind. Okay, so maybe there's something else in here that I feel pretty good about this vocab from here on in. We did a lot of amino acid work. We talked about genes, right? We're going to talk about some of the stuff next week with like enzymes. But we've talked about a lot of this stuff, right? So I'm actually good on my words. What I do know from here, right, is that it's talking about amino acids. It's talking about genes that are encoding for enzymes, right? Which I know is protein synthesis. And maybe I need to like quickly, cool. And then I'm going to get into my questions before I actually summarize some of the information. Okay, so they're giving me a circle 1 arrow that represents transcription on the template pathway. Identify the molecule that will be absent if enzyme YUC is non-functional. So they're starting to ask me about like what is transcription and what is the molecule that would be absent if this enzyme YUC is non-functional. Something else that I wanna write is that this pathway that they're showing us up here, okay, this pathway here is showing us a protein synthesis, right? Because it's showing us a gene, and then it's showing us mRNA, and then it's showing us an enzyme, which is a protein. Okay, so maybe other arrows that I think are important that you're going to have to continue to reference, right? Because it shows us tryptophan, I3PA, and IAA, right? Maybe we write that down if that's helpful. Maybe we're just like, if you feel good looking at the screen and kind of doing this with the screen, maybe you keep it here. I3 synthesize the next one, right? So two-step to enzymatic pathway. Okay. So now I have to actually go in and break down what the question is asking me. So for each of these, for A, B, and C, I'm going to fill out, okay, which is circle and then identify. And what are they actually asking us to do? Well, they're asking us to identify, so I'm going to focus on this. But if you were to answer circle one arrow that represents transcription, well, we already said it here and say, okay, well, I know a gene is DNA and mRNA is going to be the second part of this. So this would be my transcription. Okay, so that's just a content piece. Again, you're probably not going to the molecule that would be absent if enzyme YUC is non-functional. Well, if YUC doesn't work, okay, so enzyme YUC, where is it? It's right here. Well, enzyme YUC is necessary to make I3PA make ultimately IAA. Predict and justify. So let's go back and figure out what we need to predict and justify. So I need to predict how the deletion of one pair of the fourth codon of the coding region of the gene TRP, tryptophan T, would most likely affect the production of IAA, and justify your prediction. So I need to predict how the fourth codon of the coding region of this is going to affect the production of IAA, and I have to justify it. Okay, so if I delete any piece of this trip T, so I have trip T here, we would imagine that along the way, right, if this is not made, we're not making this, which means we're not making this, right? So it's going to decrease in some way, is like my first thought, is because this enzyme is not going to be able to be made, right? So if you have a deletion that's a frame shift, right, this is a mutation that's going to affect this a lot, this trip T. What was my other, the I3P thing that I want to go over is C, so explain. You're missing something, right? If you don't have this, okay, if we turn this off here, it's going to stop the production of this, okay? It's not going to promote the production of this, right? That would be, and go back to, maybe I'm writing here. So I'm going to start off with my seconds. I've already done my first step is just like reading the prompt. That's what I'm doing right now. My second step is going to be actually going through and writing down on my new piece of paper kind of those boxes that we just talked about. So I'm writing, and you don't have to draw boxes if you don't want. You can kind of do this however you want, but I think that this can be helpful. So I'm going to go ahead and say I want to look up hormone to start, okay? Words that we're writing in here, so we're not wasting a whole lot of time with this. So maybe I'm going to then write in under hormone, maybe I forgot that synthesized means to create, so I'm gonna write that. So this means to create. A hormone is a molecule that sends messages, right? So once I look that up, I'm gonna have this in here, and we're actually gonna have kind of a cheat sheet that we're gonna make before the exam, so hopefully you'll have those in front of you that can help you as well as you guys look through in my reference sheet, which we're going to start making our reference sheets in a couple weeks. But right now we can just actually like look these things up in our notes. But I'm going to write, like, maybe I need to just quickly review protein synthesis because that's the process that they're talking about. Okay. And maybe like transcription, translation. Maybe I already know this stuff, right? But you might want to just quickly look through your notes on this because this is the process that they're asking us about, okay? So some of the information that I need to make sure that I have here then is that transcription, the process is taking DNA and making it into RNA. Okay. That might be information that I got from up here, right? But that's outside information that I'm bringing in that I'm going to say. The thing that I'm going to write and I'm going to remind myself, right, is that enzymes are proteins. And that this diagram, the figure is showing us that we're going from gene to mRNA to a protein, right? And then it has some of, maybe I'm writing tryptophan to A to IAA, right? These have to be made in order to kind of start a chart underneath this. Let's do this, right? That has my letter, right, with the task verb. Okay, so circle and identify are the two task verbs that I was given. This is definitely a 1 and this is a 1. These are very low detail. So the circle is going to happen on the actual diagram. I don't imagine that they're going to have you do anything like this on the AP test that we're going to get that's digital. But identify, we know, is a 1. That just means you're writing one word. Okay. Transcription is going to be going from... transcription is going to be going from where's this? DNA, right? DNA to mRNA. So let's use that information that maybe I looked at a question like that you have to draw on a diagram on this digital exam. Okay, so I just like knew this answer. I'm just going to write IAA in here. Okay. Then I'm going to do my 2 task verbs. Okay, so I'm going to go ahead and I'm going to move up all my stuff. Okay, I'm going to make a line in between these 2 things. Okay, and I'm going to write predict and justify, which we know. Okay, predict is a 1, justify is usually a 3, okay, in terms of detail. So remember, predict is usually going to be decrease or stay the same or increase. So I'm going to write for my predict... IAA will, you could say production won't happen, right? Stop. You could say it would decrease, right? And the reason for that, I'm going to go ahead and say that like my trip T isn't made. These are just like ideas I'm having, right? Isn't made, so cannot be an enzyme to make which means that we're not going to make our IAA. So these are just notes right now, right? You guys can see that I'm kind of just jotting notes. I want you to be jotting notes as you're doing this, and then if you have time, you will write these out and actually like verify, edit your answers a little bit. But if you do not finish, you are just going to take a picture of this. So I actually want you guys to take a picture of just this and upload it if you don't have time. Okay, so this information will get a picture and you'll upload it if you don't have time to rewrite. Okay, so we have not talked about feedback mechanisms yet. So I really just want to talk about my task verb explain here. So my explain is going to be a 3. And it's asking me to explain what the feedback is that's here. Okay, so if I were in there's only 2 types of feedback, positive and negative, right? So a lot of you actually guessed this correct. My initial thought maybe is negative feedback. And it should be negative feedback because feedback. And then remember, when we're asked to explain, we need to both ID, which would be negative feedback, and then our explanation needs to actually say like why. What is this negative feedback? I do want, if we look at the rubric, what you needed to have here, okay, is that you are going to have in C, which is down here, okay, you need to say that negative feedback or feedback inhibition, right, will increase the amounts of IAA, which will inhibit the pathway, okay, or that the production of YUC enzyme is inhibited, or the YUC activity is inhibited. So you needed to say a couple things. Yes, you had to identify this, which is going to increase the IAA. And then you also have the production of YUC enzyme is inhibited, or that the YUC enzyme is inhibited. Okay, so the production of this or the actual enzyme activity is inhibited. Remember, inhibited means to stop. Okay, cool. Great. And then you can see some of the other, you can pause this and look at this if there was something that was confusing to you in terms of content here. But this is the rubric for number 1, and everything that you needed to include. Specifically, this idea of like the mutations resulting in translation of an inactive or a non-functional TRPT enzyme. So remember, we're saying that TRPT is not going to be made, right? And that's going to make it so that we have this reduced production or no production of IA, right? So what's important to note here is that we know that mutations, if we have a frameshift mutation, that we're going to have it's going to affect the enzyme in some way, because a frameshift, a deletion, is going to affect this a lot. So it's going to result either in no translation, it's going to make an inactive or non-functional enzyme, or it's not going to make I3PA is also what you could have said, right? So that's this one. You could have said like, because we know that this isn't working, this isn't going to work. I do want to note this one piece right here that says the mutation will result in the translation of a non-functional or inactive enzyme. Okay, remember this is a protein. Guys, you cannot say that yes, structure determines function, and that's great. I'm glad that we have all thought that, right? But the thing that we all wrote is that the function is going to change. It's not that it's going to change and become a new protein, right? It's that the function changes, but specifically the way it changes, okay? It's not going to have a new function, right? It just doesn't work. Okay, so that's important to note. It's not like miraculously we get this new protein. It is possible that, like, think about sickle cell anemia, right? Sickle cell anemia is going to make a protein that it actually still makes the protein, right? That missense mutation, the one amino acid that changes, but it's not making a new protein. It's making a protein that doesn't work as well, right? The hemoglobin is just not as, it's messing up the shape of the cell, and it's not able to move through your body as well. So it's either making something that's non-functional, or it's not as effective. Okay, so I want to make sure that was clear in terms of content. Okay, that is the end of this video.